Okay, this is part 2. In part 1, we looked over the nucleus that houses your DNA and the plasmic reticulum, which is a parking lot for these small white dots, ribosomes, organelles that synthesize protein. We also look at the Golgi apparatus right here, the organelle that gets all the proteins from endoplasmic reticulum, modifies them, like post office puts some addresses and tags on all of the packages, and then send them out in these little vesicles, protein vehicles, to go to different parts of the cell or outside of the cell. We also learned that um, the part of endoplasmic reticulum which is not rough but smooth synthesizes lipids, and we learned about the role of mitochondria in our longevity and the fact that it sucks the energy of chemical bonds of glucose and carbohydrates into the special energy carrier of the cell that is called ATP. Uh, well, what else we have left? We have left peroxisomes, which are usually positioned close to the mitochondria and deal with those free radicals that mitochondria create plenty of. And another thing interesting for us is so-called lysosome. Lysosome is a vesicle, similar to vesicle, but the proteins that are in the lysosome are special lysous proteins. It means they're killing proteins. Lysosome, if it sees a virus in the cell or part of bacteria or something foreign, it comes to this place, engulfs the foreigner, the antigen, and devours it. It also devours old organelles so the cell can build new ones. Now, what do we see? What do we see in the nucleus is nucleolus. Nucleolus is not actual organelle, it's just the place in the nucleus where your DNA is being actively transcribed into RNA. You know, DNA is located only in the nucleus, protein is synthesized on outside of the nucleus. We need some instructions coming from the nucleus to the cytoplasm. Those instructions are copies of DNA called RNA. This RNA is used not only for instructions but also for making ribosomes. That's why ribosome is called ribo and some, and RNA is ribonucleic acid. So this nucleolus, it's usually darker than the nucleus, is the place where DNA is being actively transcribed to make more rRNA. And plasma membrane. Here you see the last organelle, the second to last. It's nice and smooth on this model, but in reality, plasma membrane is covered with cell markers and receptors and gates that allow the stuff in and out of the cell. In the meantime, the stuff that gets in and out of the cell is being checked. Is the stuff is okay, or should we send it back? Plasma membrane is covered with cell markers, like for example your blood type. That's for blood cells. The other cells have other cell markers, and your immune system knows pretty much which is your cell and which is not your cell. That's why, because of those cell markers located on plasma membrane, your organ transplant or tissue transplant could be like rejected. You need to suppress immune system to make it blind to the cell markers, not to react to the cell markers, so a transplant can have an easier time to get adjusted in the new body. And there is a small organelles, centrioles. They used in cell division only, so if you haven't mentioned it on your drawing, it's okay. The organelles that needs to be mentioned are nucleus, you can mention nucleolus, endoplasmic reticulum, rough and smooth, ribosomes sitting on endoplasmic reticulum, Golgi apparatus that processes the proteins, mitochondria that create energy for the cell. What else? Lysosome, that is traveling trash station of the cell and also recycling facility filled with digestive enzyme. Uh, did I mention peroxisomes that takes care of the free radicals located near mitochondria? And vesicles filled with proteins going in and out of the cell. Okay, I hope you like this video, and if you don't, send me something saying why. I will try to provide like feedback, instructions when you need, and I just would like to know what will work the best for you. Thank you very much. Bye.